What's up, biology students? It's me again. In today's video, we're going to talk about the genetic change that occurs in a population as a result of the evolutionary process. Every population contains genetic diversity. No two individuals in any population are exactly alike because no two individuals have exactly the same genes and genetic sequences. This is true for every population of biological organisms. Every population contains variations that are more or less fit for that particular environment than other variations. Birds, bats, trees, flowers, every population, every single one, contains genetic diversity. To really understand evolution, we need to think of populations as collections of genes. This collection of genes is called a gene pool, all the genes and all the alleles for those genes that are present in a population. For example, suppose we have a population of 25 mice. 36% are brown and 64% are black. Remember, these kinds of traits are controlled by genes encoded in the DNA of these mice. The fur color gene for these mice has two alleles, black and brown. Black is dominant. That's why both the mice with the homozygous black genotype and the mice with the heterozygous genotype have black fur. But only the homozygous brown mice have brown fur. Brown is the recessive allele, so mice need two copies of this allele in order to have brown fur. If we consider only this one trait, and this one gene out of the many thousands of genes possessed by these mice, then their gene pool would look like this. Since each mouse has two fur color alleles, and since the population contains 25 mice, that means we have 50 total alleles in this gene pool. 30 of them are brown, and 20 are black. That's 60% brown alleles and 40% black alleles. You should notice in this gene pool kind of view that despite the fact that only 36% of the population actually has brown fur, 60% of the alleles in this population are brown alleles. The difference is due to the fact that a single brown allele may be hidden from view by the presence of only a single dominant black allele. So in many ways, the gene pool view of a population is more informative, even if we can't see the specific individuals who carry those genes. Fur color in those mice is a single gene trait, meaning that this trait is controlled by only a single gene. Most of our traits, however, are determined by several genes, not just one gene, and we call these polygenic traits. Eye color, for example, is determined by at least two genes. This chart shows us which combinations result in brown, green, or blue-eyed phenotypes based on alleles for both of these genes. As you can see, more combinations result in the brown phenotype than any other color, but both the HERC2 gene and the GAY gene influence the phenotype of the individual. All populations contain genetic variation. No gene pool is completely uniform throughout. There are several reasons why this is so. In organisms that reproduce sexually, the fact that chromosomes are independently assorted during meiosis means that many possible combinations of genes could end up in the resulting gametes. In humans, who have 23 pairs of chromosomes, that would be 2 to the power of 23, or about 8.4 million different genetic combinations in the gametes of just a single individual. And that's not even accounting for the recombination that occurs during crossing over, which also happens during meiosis, and which further shuffles the genetic deck. Crossing over, remember, is when homologous chromosomes sort of swap genes, so genes from one chromosome end up on the other chromosome. This means even more possible genetic combinations and more genetic variation in a population as a result. Sexual reproduction produces variation because it requires the fusion of two gametes from two individuals, each of whom is capable of producing many possible genetic combinations in their gametes. And sexual reproduction isn't even the only source of genetic variation in a population. Random mutations are another source of genetic variation within a population. A mutation is any change in the genetic sequence of an individual, and most of these changes are neutral and do not produce new or modified traits. They do, however, provide an important window into evolutionary history. Neutral mutations are important for interpreting what's called the molecular clock. The molecular clock uses the laws of probability and the average rate at which neutral mutations occur in order to figure out how long a particular species has been evolving. Neutral mutations are the ticks of this clock because they tend to occur at a pretty constant rate. For example, if we have a sequence of DNA that is not being expressed, an intron maybe, we can look at the differences in this sequence that exist in related species and figure out what the original sequence must have been and how long it took for these mutations or differences to accumulate. This graph shows how long it has been since different species shared a common ancestor according to the molecular clock. And remember, we're looking at differences in their introns based on neutral mutations that really aren't affecting their health or fitness. Distantly related species would share a much older common ancestor, while closely related species would share a more recent common ancestor. The molecular clock data supports this idea, as we can see here. Horses and donkeys, two very similar species, share a common ancestor that lived only a few million years ago according to the molecular clock. But humans and kangaroos, who are much more different from one another than horses and donkeys, share a common ancestor that lived almost 125 million years ago according to the molecular clock. But not all mutations are neutral, and non-neutral mutations are a major source of new alleles and new traits in a population. Sometimes it happens like this. A replication error causes there to be a duplicated copy of a gene. If that in and of itself doesn't change anything, then suppose a mutation occurs in one of these two duplicated genes, and this results in a new function or usefulness. The original gene with its original function is still present, but now a new allele exists in this population. And if it confers some kind of advantage, that could result in the evolution of this population towards one in which that new allele and that new trait for which it codes are more common. Of course, some mutations could be harmful as well, in which case the carriers of these alleles would be at a disadvantage. Some mutations produce bigger changes than others. Earlier in the year, we talked about homeotic genes, the master control genes that guide the development of an organism. You might also recall that all animals have a fundamentally similar set of homeotic genes that we call homeobox or Hox genes. All animals have these Hox genes, but we differ in how many copies of each we have. And these differences are a big part of why different groups of animals, like flies and fish, look so different even though their development is controlled by a similar set of Hox genes. Because these genes guide development, changes to these genes cause pretty major changes in development, meaning possibly big changes in the overall form and body plan of an organism. Experiments have been done in the lab in which the Hox genes of fruit flies have been forced to mutate. And the result could be something like growing a leg where an antenna should be by putting this gene for leg development in this spot that would normally code for head development, or growing an extra set of wings by duplicating this gene that guides the development of the thorax. Legs where antennas should be might not be such an advantage, but you can see how an extra set of wings might increase an organism's fitness. These kinds of changes to homeotic genes play a major role in some of the big shifts in the evolution of animals. For example, we think that a mutation to the genes that control leg development in a very ancient lineage that predates modern insects by about 400 million years could explain why modern insects only have 6 legs when similar organisms in the fossil record had many more legs. Something about this change must have been advantageous because 6-legged insects are quite common today. Our modern definition of evolution includes the idea that changes to the gene pool of a population are the result of the evolutionary process. Certain alleles become more common, while others become less common. And this happens because the carriers of those alleles differ in their fitness, that is, their ability to survive and reproduce in a particular environment. Of course, if we understand the relationship between genes and traits, this should make sense. Genes give an organism its traits, so it is these genes that determine the fitness of an organism in its environment. If an organism has low fitness, its genes might be removed from the population, removed from the gene pool. And if an organism has high fitness, then it is likely that its genes will be passed on to the next generation and will become more common in the gene pool over time. In our example here, the alleles that make these beetles green confers a disadvantage because birds prefer to eat the green beetles. Maybe they taste better, or maybe birds can simply spot them easier and catch them more frequently as a result. Whatever the case, the selective pressure exerted by the birds will cause significant changes in the gene pool of these beetles. the green allele will become less common because of the disadvantage it confers, and the brown allele will become more common because having it means that the beetle is more likely to survive and pass that allele on to its offspring. So natural selection has the capacity to alter the gene pool of a population, resulting in evolutionary changes over generations. Natural selection can affect the gene pool of a population in one of several ways. Because most of our traits are controlled by several genes, that means a lot of variation in phenotypes. Think of human height, for example. Humans don't just come in two heights like our beetles came in two colors. Humans come in all sizes from very short to very tall and everywhere in between. And this is typical of many traits in living organisms. In this example, we're looking at fur color in a different species, and in this case, fur color must be controlled by several genes because this species ranges from light-colored fur to dark-colored fur with everything in between. If this graph represents our original population, we notice that most individuals have a phenotype somewhere towards the middle of the spectrum, medium brown, if you will. Fewer individuals have the lighter and darker phenotypes, and that's why the graph drops down low on this side and on this side. The height of this line represents the number of individuals in the population with a particular phenotype. And since phenotype is controlled by your genes, the line pretty much also represents the number of individuals in a population that have a particular genotype as well. And we know, for example, that the allele combinations that produce this middle brown phenotype are the most common, while lighter and darker brown allele combinations are less common. Depending on which of these alleles are most advantageous, one of three things could happen. If the alleles that make this species very light or very dark confer a disadvantage compared to the middle brown phenotype, then those extreme phenotypes will become less common and the middle phenotype will become more common. This is called stabilizing selection, and the results are depicted here in this graph. It's like an evolutionary push towards the middle. If alleles that make this species dark in color are advantageous, then the darker phenotypes will become common, while the lighter phenotypes become less common. This is called directional selection, and the results are depicted here in this graph. Think of this like an evolutionary push in one direction. If alleles at opposite extremes are advantageous, either very light or very dark, while alleles that result in a middle phenotype are not as advantageous, then the middle phenotype will become less common, while both the very light and the very dark phenotypes and their associated alleles will become more common. This is called disruptive selection, and the results are depicted here in this graph. This is like an evolutionary push in two different and opposite directions. Human birth weight is a good example of a trait influenced by stabilizing selection. For example, babies born with very low birth weight are more vulnerable to complications and are more likely to die in infancy as a result. Babies with high birth weight are also more vulnerable to complications, as are the mothers who carry them and give birth to them, so this is also a disadvantage. Babies with a middle range birth weight have the best chance of survival, so these babies are more likely to grow up and reproduce themselves someday, in which case they will pass the genes that gave them a middle range birth weight on to their children. This is an example of stabilizing selection, an evolutionary push towards the middle. Our giraffe example is a good example of directional selection, as was the process that led to the evolution of the modern horse. That is, if you're thinking about both of these traits in the Darwinian sense and not the Lamarckian sense, because remember, giraffes didn't get a longer neck simply by stretching. Since giraffes with short necks are less likely to survive because it's more difficult for them to obtain enough food, they pass their short neck genes on less frequently than giraffes with long necks. This pattern causes giraffe necks to gradually become longer with each successive generation. This is an evolutionary push in the direction of longer necks. Modern horses evolved a larger body size for similar reasons. Naturally bigger individuals were more likely to survive and reproduce, so their genes became more common in the population. An evolutionary push in the direction of a larger body size. Natural selection acting on the body size of the coho salmon, however, is a good example of disruptive selection. In this case, the advantage goes to those salmons who are either small or large, but it is actually a disadvantage to be of a medium size. Large male coho salmon outcompete medium-sized males for territory, but the small-sized males are able to be sneaky because of their small size, and are often able to reproduce by using their small size to stay out of sight of the large territorial males. So, the medium-sized coho salmon, and the genes that make them this way, become less common, while small and large coho salmon, with their small and large genes, both become more common, an evolutionary push in opposite directions. Interestingly, natural selection isn't the only thing that causes changes in the frequency of alleles in the gene pool. A random change to the gene pool not associated with natural selection is called genetic drift. One way this can happen is due to the founder effect. For example, say that we were to transport a small handful of beetles into a new environment. Depending on which beetles we randomly grabbed from the original population, we may form a new population with very different gene pools than the original. In the first example, we just so happen to randomly pick yellow and black beetles, and no red beetles, so the yellow and black alleles will be the only ones in the new gene pool, and there will not be any red alleles at all. Likewise, if we randomly picked these beetles from the original population, we'd only be taking red and yellow alleles to form our new gene pool, and there wouldn't be any black alleles. These two populations are likely to evolve differently over time based on the gene pool from which their population began. And the important thing in this example is that it was random. We didn't choose beetles based on their fitness, we simply picked beetles at random to start a new population. Similar things happen when plants and animals are accidentally transported into new environments, say by high winds during a storm or through human shipping of goods across the globe. Another example of genetic drift is called the bottleneck effect, and it is very similar to the founder effect in many ways. In this case, it has less to do with moving into new environments, which is what the founder effect is all about, and more to do with some extreme natural disaster that wipes out a significant portion of the population, regardless of fitness. Because even extremely fit animals can be killed during a tornado, while less fit organisms may survive by chance simply because they happen to be in the right place at the right time. But the next generation is always produced by the survivors, and if the surviving population has a different gene pool than the original population, because of the random chance of some natural disaster taking out a huge percentage of that population, that will affect the future evolution of this population. So with all that's going on in a real environment, are populations ever not evolving? One pair of scientists, Hardy and Weinberg, came up with a theory that addresses this very question. Hardy-Weinberg Equilibrium is a theoretical state of stability and unchangingness. This is based on a set of conditions that must be met in order for a gene pool to remain stable and unchanging over time. And if the gene pool stays the same over time, that means that no evolution is occurring. The conditions necessary to maintain a stable, unchanging gene pool are as follows. And remember, all 5 of these criteria must be met in order for the gene pool to remain stable. 1. Very large populations, so large that genetic drift can't really affect them. 2. No emigration or immigration. No new members enter the population, and none are lost. This doesn't count natural birth and death. It really just means that no foreign genes can come in, and no genes can leave the population to go somewhere else. 3. No mutations, because mutations would potentially result in new alleles, and that would cause a gene pool shift. 4. Random mating. No sexual selection. Every individual has an equal chance of mating and passing on their genes. And 5. No natural selection. All variations contribute equally to the fitness of organisms in that population, and every individual has an equal chance of survival. If all 5 conditions are met, then theoretically the population would never evolve and its gene pool would never change. Remember, Alleles don't suddenly appear into existence out of nowhere, and since they are passed from one generation of organisms to the next, and each new generation is simply a continuation of the previous generation's genes, we would always find the same frequency of alleles generation after generation because nothing could possibly cause them to change. The alleles in the pool are there and always will be, if our population meets all 5 of these conditions. But ask yourself, is it really possible for an actual population to meet all 5 of these conditions all the time? No way, not a chance. And even if one of these conditions is not met, it means that alleles could be added or removed from the gene pool, and this could result in survival advantages for some individuals, and there begins the process of evolution, even if it may proceed incredibly slowly. What's the point? Why bother with a theory for something that could never actually occur? Well, as scientists, we always need something to compare our observations against, and to test our predictions against. Hardy-Weinberg Equilibrium provides that basis for comparison, the baseline against which we can measure evolutionary change. There's even a mathematical side to this Hardy-Weinberg Equilibrium. In this equation, P and Q are variables that represent the frequency of the dominant and the recessive allele for a particular trait, respectively. So P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. P plus Q always equals 1, because that represents 100% of the alleles in a population. So if 75% of the alleles in the gene pool of a population were recessive alleles, Q would be equal to 0.75, and P would have to be equal to 0.25, or 25% of the gene pool. So, if we know what the gene pool looks like for one generation, we can predict what the next generation will look like mathematically, providing that the 5 conditions for maintaining equilibrium are met. P squared equals the frequency of the homozygous dominant genotype, or the proportion of individuals with 2 dominant alleles, in the next generation, given our original numbers for P and Q. Q squared equals the frequency of the homozygous recessive genotype, or the proportion of individuals with 2 recessive alleles in the next generation, based once again on our original numbers. And finally, 2PQ equals the frequency of the heterozygous genotype in the next generation. This is what we would predict to find if no evolutionary changes were occurring in this population, if all 5 of those conditions were met. Then, if we measure the next generation and find that the reality does not match our mathematical predictions, then we know that the population is evolving because their gene pool is changing. Evolutionary change is inevitable because it is nearly impossible to keep the gene pool of a population from changing in some way over time. This gene pool view of a population helps us to assess the rate and extent to which evolution is occurring in this population, and broadens our definition of evolution to include any changes in the genetic makeup of a population over time. And a thorough understanding of evolution involves knowing how natural selection changes the gene pool of a population. This gene pool is essentially the bank of all traits possessed by a population, and all members of a population withdraw their individual traits from those available in the gene pool, so that when the gene pool changes, the population changes as well. It's all part of the evolutionary process. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand what this gene pool business is all about, and how gene pools may change due to natural selection and genetic drift.